G'day, good morning, good evening. Hello, I hope you're well when you listen. Thanks for listening. This is a medical cannabis show, and I respect the law, and I hope you do too. Today is an open topic, so I haven't decided what we're going to discuss yet. I hope the viewers can decide. That would be good, and then make a good show of it. So if you're re-listening back to this, and watching, I suppose, because there'll be slides later on, hopefully. Skip forward probably 5 to 10 minutes, at least, something like that, until it gets into until you can see some sort of slides that come through, is my suggestion, unless you want to hear me ramble, because, I like to get the topic, and then we can sort of focus on the topic and elaborate. I can show all the slides and other references and see what sort of experience I've had in that area. So I'll give it till 2 minutes, just to pause and I'll start talking, then wait for people to arrive, I hope. Like Lene. Hello, Lene. Looks like the teacher is tardy. I don't know what tardy means. Or maybe you're early. Yes, you're early from the time. Hello. That was sent 15 minutes ago. You can see the time there. But it's nice to see you. Thanks for listening. And if there's any topics you'd like to discuss, please put them in chat. So that's how we can start. All right, the 2 minutes has been up. So what am I going to talk about while I wait for some people to arrive? Well, what can I show? I haven't talked about microbes for a while. That's always interesting. And beneficials, how to keep them. So microbes. Until so please put what? How? All right, sharing screen. Microbes. Different types of microbes. Beneficials. Let's just start and see, because I don't know what really to discuss. So please put your questions into chat. Of all things, talking about a bit of the human stuff. Actually, I'll just skip through that because, what's this? A trap. So this is a light trap. So see, it's solar powered on top, and there's light in the bottom, and they put some sort of sticky substance to trap it. And yeah, that's an example of how to trap. If you've got pest problems, I suppose, it's microbe related. That's the algae format, breakdown, different types. When you get your seaweed emulsion and kelp meal and stuff like that, there's usually two different types. They've got the brown and the green algae, and one's from different hemispheres. Oh, gee whiz. I can't remember, but having the two different types helps because you just get a little bit of mixture. That's really good organic feed, that is. What's this one? Cyanobacteria. How they produce. They change nitrogen from the atmosphere with nitrogenase into ammonia. So if you have some cyanobacteria, you can get some free ammonia by using that biofertilizer, it's called. And I use Athrospiroplatensis cyanobacteria because I want some free nitrogen. So many types of pollutants. Different ways to get a plant polluted, because it's a bioaccumulator, so all these things can accumulate. So if it's grown in a bad environment, even near a factory, stuff like that, it's going to accumulate it in the different parts of the plant And if you were to smoke those different parts where it's accumulated in, you're going to get that little bit. All sorts of bad things. It's how Mother Nature's cycles. Otherwise, there'd be too much of a build-up everywhere. All right, don't really see much of it. I've got my tablet going as well, hoping to see some questions come up from different people, but it's not really much at the moment. So I'll go a little 10 minutes. Then if there's not much after 10 minutes, I'll stop it. The duration of the effects. Oh, righto. I don't need to talk about that one. Pathogens. Oh, yeah, this is their pathways for different pathogen, the responses. So let's not get into that too much. The reason. No. Oh, that's just how. What's this? Lactic acid bacteria. Labs are able to grow in presence of air, even though they have lack catalase and the enzyme, because they contain oxidase, which is an enzyme which catalyzes the oxidation of organic compounds like peroxide, hydrogen peroxide. Most microbes belong to this category. I'm going to get to something interesting that relates a bit better. Removal of infected leaves. Yeah, well, whenever you're looking at it, you should always turn it upside down, because most things are laid on the undersides of the leaves. So that's, if you can pluck some off, always turn it over, because you might be surprised. And just because the dot's there doesn't mean that they're harmful ones, because they could be good stuff too. See, this says, look, pest-free. Looks just showing nothing, but that would be an inside conditions. Where if it was outside, there should be at least a few dots on it, but they should be beneficial ones. Because the harmful ones, they'll form a hypersensitive reaction around it where it'll kill the cells in it. And you'll see like dots starting to form on the surface of it. Oh, here you go, some biopesticides. So I hope everybody's into the biopesticide, the organic way, because you can nearly treat most problems through biopesticides. And this chart's a good way to start. So for instance, if you've got, what's an easy one? What's a common one? So some of these like sucking insects. That'd be like phloem feeders. So you might get an example of those are phloem feeders, like aphids are. So if you've got aphids, you just get ladybugs, and that would say it here somewhere. So that's just the example of what it is. That's another good one. A disease. It's Pythium. Rot. Just got it over here. It says it in it. Glad. Okay. This doesn't show exactly what it's for. This just shows the beneficial ones which help in those areas. So it doesn't say if you've got rot, you should go and put in Pseudomonas, which it's actually Pythium. Rot. Now let's go on to another one. Oh, yuck, you don't want to see that. Look at that sad looking thing. Leaf miners too, you'll see little lines under it, like wiggled lines in the top, because they burrow in between the tissues. Yeah, all right. Most of this stuff's pretty similar to get rid of. Thrips are hard at her. Leaf hoppers or plant hoppers. Long skinny looking insects. Spider mites. Yeah, a lot of these is your environment, like the powdery mildew. Seasonal changes. Fungus gnats could be in your substrate that you buy. Eggs and that. That's pretty cool. This is about quorum sensing and how bacteria communicate. So you know how bacteria put out a big biofilm, like you'll see it, that slimy stuff around different microbes, little areas, that's the area that they protect themselves in. So they've got in that antibiotics, which tells other microbes to go away because they want to hog the area. So it's pretty fascinating. They're good, and they just keep multiplying if they're in their favourable conditions, like this is doing through horizontal gene transfer. Yeah, the quorum sensing is pretty amazing. They speak and they put out these little chemical sensors just to ward off, and they can speak within each other, and they can tell others to go away. Antibiotic vancomycin. Let's get something a bit more interesting, embracing. That's more about the organic microbe-riddled land and substrate to one that's not, to persons that's poison it and synthetically feed. So you'll get all sorts of build-up salt. You put it in, the salts will build up. They won't be able to filter properly. Microbes won't be able to cycle properly, and just problematic. That's why the planet's getting bad. So yeah, that's why I only promote sustainable organics, because it does it keeps doing on the left, and you get pretty much what you can get in synthetics. You get a lot better secondary metabolite profile. That's blooming sure. Effective drought stress on soils. Righto. These other beneficials. Unveiling the hidden heroes in plant health and ecosystem sustainably. In 3 different areas of the plant. So the rhizosphere is right next to the roots. The endosphere is inside of the plant, and the phyllosphere is from the ground upwards. It's the 3 different areas, and there's heaps of different groups and like cultivars and genuses, not cultivars. Sorry. Double drink. Yes, there's. I don't even know what I was saying. Oh, that's right. The ones that are in the plants, there's not just a few different types, there's many. This is what microbes need to survive. So if you want, I've done a fair few culturing courses. That's how to test your microbes. If you want to see if they've got what it says on your packet, so it'll say on the packet times Bacillus subtilis times 10 to the power of 5. So you go, all right, what's 5 mean? Well, that means if you dilute serial dilution 5 times, 1, 2, 3, 4, 5, you should have their number. So they'll say 4 to the power of 4 times 10 to the power of 5, which means if you serial dilute it 5 times, you should have 4 dots in your Petri dish. So you can test their numbers. Make sure you're getting what you pay for, because they're in some seed form, the endospores, the microbes. So the reason why you buy them dry and then you put water in it, because it brings them back to their favorable conditions and they germinate. Because half the might say if they look like a tic tac, half of it is a seed, an endosphere, which is this thing. Which is pretty cool. What's this thing say? It's a little cool wheel. The Bacillus genus. There's heaps of beneficial ones in that. There's subtilis, which is very common. Bacillus thuringiensis, BT. Anyway, what's the ideal method for growing? Okay, we won't go into that. Russet mites. That's a little cool electroscanning microscope of one. It looks pretty big, doesn't it? You can't even see them under a microscope. What's this? Azospirillum. Oh, yeah. Azospirillum is a beneficial that's a non-symbiotic nitrogen-fixing bacteria. That means that it doesn't have to be symbiotic with a plant, like rhizobium has to be symbiotic with a legume for it to convert nitrogen into another form of nitrogen, where azospirillum and azotobacter, they do just that. I use azotobacter. Some protozoa, it's how to culture some. For primo MedCanna. I like that because it's just like it's got the micros. I suppose I should. Oh, yep, Shipwreck's arrived as well. Hello, Shipwreck. Nice to see you. Just talking about microbes at the moment. It's really going through a few slides. I'm not really sure what to talk about. So if anybody's got any questions, please drop them. So I like microbes. If you can get good colonies and good food cycle web happening, you can't lose. It'll take care of itself. So I suppose I'll just talk a bit about this, another few more minutes, and that'll be it. Unless any questions come through. I'll try and keep it related to the MedCanna stuff too. That's how to identify them under the microscope, by their shapes. Won't go into it. That's me testing some Rhizophagus irregularis on a plate. This is very awesome. If you've ever got any problems, pretty much 2 biopesticides can take care of it. Montgenesis, this one here, which unfortunately is hard to get in North America, and lacewings. Look up the top. So if you've got any of these problems, you can pretty much get rid of them with those 2 things. So if you're in winter and you're coming into spring, you might want to consider spending 35 and lacewings, 140 odd bucks delivered to get yourself a couple of canisters of biopesticides. It might save you. It'll save you spraying, especially if you're organic, and if you renew your reuse your substrates or put them back through a cycling of compost or something like that. These microbes will stay in your substrates, and you can keep them forever. So it's so beneficial. Yeah, that's pretty cool, isn't it? The nitrogen cycle. So you get all different types of microbes. For this nitrogen cycle to happen, it's pretty much microbes. So they've got to react with what's that one that takes like the azotobacter, which takes nitrogen and N2 and converts it into NH3. So right here would be your azotobacter. So you need microbes to really, really help with these processes. Or you can use synthetics and just put in the 2 available nitrogen forms of the plant, which is nitrate and NH4, which isn't here, ammonium. So that's why microbes are so important. Nitrosomus. Now, what was the other one I was thinking of? It's not there. Different, that's terminology. They'd probably bore you. Transfers of different things. Microbes also need micronutrients to perform certain metabolic functions too. So yeah, really, with your feeding, with organics, you should be feeding your microbes. Like vitamin B1, thiamine, is so beneficial for the metabolic processes. So I include thiamine, and you can go and buy it in those hydroponic shops, but they've also put in other synthetic things, like they'll add some nitrogen, some potassium possibly, and yeah, so just go and buy pure thiamine and then dilute it back to the ratio. They put in, can you believe, into those formulas that they make, it's 0.25% is B vitamins. So if you go and get some hydroponic store that says, all right, it says B vitamins on the front, turn on the back and look at the percent. It's under 1%. So you can go and buy your own bottle and make your own for I don't know, take a few zeros off the price. And then you can get into your organics as well. You can eliminate your, but that's how an example of micronutrients. What's this? Oh, this is how they reduce pollutants. Okay. It's getting into a bit of detail. How else are they important? What else can different areas, processes? Is it azo? There's azotobacter, electron acceptor, oxygen, and this electron donor is nitrogen. So it will use oxygen and nitrogen to perform its processes, and it will put out NH3. All right, so ammonium is good because that's one of the ones. NH3 is a gas, and you need another microbe to convert it into NH4, which is nitrosomes. That's what I was thinking of before, and then that's one of the plant available forms, because only these two here is the plant available form of nitrogen that it can accept. If it comes, it's sitting there in any other form, in these forms here, dinitrogen, nitrogen, ammonia, any of those, it just won't take it up because it's not the right form. There's nitrosomes. Yeah, see, NH4. It converts the ammonia to ammonium. All right. This hasn't been much. I was going to relate it. Right at the end is a cool, because I've shown this before, is the chart of the plant where it shows all the different microbes at the different stages. And there's over, I don't know, it's probably at least 100 or so. That's how you can get, if you want if you can't afford your azotobacter, you can get it yourself by doing this. I've never done this, but this is the way that you can do it. So if you like to play around with your science stuff, yeah, grab your mix. And then the reason why you want to put it in is because the plate without ammonium will be the plate that it will work on, because it's going to produce a lot in that substrate. Because if you're going and giving it to it, it's not going to produce anything. So that's why it's the plate that's the winner. Yeah, most of the world's 99.9% of the world's microbes are undiscovered yet because they're unculturable. Isn't that amazing? Oh, there's a chart up here. Yeah, here you go. Look at that in the ocean. 0.1% is the maximum that's been cultured in the ocean. 0.1. In the sludges, there's a lot more, because in our sewage and wastes, that's where they've done lots of studies. It's getting into the anaerobics stage where it's starting to produce harmful ones. E. coli. You know, E. coli, if it's in its favourable conditions, it can spread around the planet within 48 hours. It can happen really, really fast. It's another serial dilution. And if you want to count microbes too, you get a hemacytometer. It's a microslide there. It's the glass that goes on top of it, the cover slip, and on the cover slip, or you could even get the one that's big and bulky, and it's got lines on it, and they're at a microscopic little level. So when you look through your microscope, it just looks like a grid pattern, and then you can count how many dots are under that. So after you've done your serial dilution, you get it down to a gridding pattern, and you put that under the microscope and you can count them. What other microbe stuff? I want to get to that cool chart. Oh, actually, this is really rad. In your substrate, particularly soil, if you've got clumpy soil and it could be anaerobic in the middle, producing those pathogens, Umycetes and Pythium and Fusarium, and the ones that Fusarium. It's not Fusarium. Anyway, the bad Umycetes that you don't want, because it can. These are the stratified layers here, where you can see the air that comes into it, and because it's so clumpy in the center, particularly clay, it could be anaerobic in the middle. So that's, if I ever see anything, rule of thumb, that's 1/2 inch across or 12 millimeters across, I'll always just squash it if it's bigger than that, because this is pretty. Yeah, that was an eye opener for me. That's from Brock's Book of Biology. Microbe. It's there's a 15th edition now, and that's my microbe teacher said, if you ever want anything fascinating, get that. And I thought, oh, whatever, yeah, righto. It is fascinating. It really is. It's most of the class notes came from those books from Brock's Biology. You need favorable conditions for microbes to grow, for them to multiply. That's why when you're in flower, if you get a higher humidity, that's entering the favorable conditions, because they need moisture levels at a certain amount, above 65%, particularly 75, and you're in dangerous areas. And remember, that's just not in the air. That might be in the air, but you imagine if you've got a big chunky number, inside of that it might be 85, 90%, which is a level where bacteria will start, and that's why you're going to get bud rot. So by not meeting their favorable conditions, you don't allow that process to happen. Yes, plant growth promoting rhizobacteria. This is biopesticide, biofertilizers, different types of them. They're just the winner, because once you get it into your soil, into your substrates, they're there forever. They can do so many different beneficial things, and then you let them sort themselves out, because in good substrates there is a even amount of pathogens to what there is beneficials. All these ones really help. That's not in. Oh, this is a nitrogen fix. That's where you should use the symbiotic one, like azos, because other ones require hosts to live on different plants. So yeah, it's free nitrogen. And nitrogen, remember, of the 17 essential minerals that a plant needs, nitrogen is the fourth one of 1.5%. Because the first three are CH and O, carbon, hydrogen, and oxygen, which you get it from the air, which make up 95%. And the next one down is in nitrogen of 1.5%. So if you can get that also from the air through microbes processing it, you've taken care of 97.5% of the plant's essential elements through the air, if you can get the right cycling happening, which I try and encourage and promote. It's free fertilizer. And some of them, like, they've got hormones in them, phytohormones, like is it Fusarium? No, it's there's a fella in Japan who discovered the microbe and named it something gibberellins, and Fusarium gibberellins or something like that, and that's the one that produces gibberellic acid. So if you can get that microbe, you're getting free phytohormones, which all these do different things, beneficials at different growth stages. So you can get it all for free. It's such a win-win scenario, organics, if you can get it happening. It's just a little bit more labor intensive, but look at the end result. See how this planet survived to this date. I don't see any more questions on my tablet. I hope there's no more. Phytohormones. I'll just talk about this for a bit more. The root exudates. They're the things that the chemicals that the root exudes or puts out, and microbes pick up on that and can give it what it wants, can kind of answer it. So they also put out protons, hydrogen plus, and they need something to exchange that with in the cation exchange, so that can exchange elements. So calcium can come in, other things can come in. So through microbe interactions, it's so beneficial. There's the rhizosphere, and then the rhizoplane is right on the edge of it. Inside the endosphere. But they also process inside. See, every single section has different microbes in it. Free living, azos, they're the ones that you want. Non-symbiotic, they're the ones that just thrive with the plant. You can't go wrong. It's azos particular. Different types of bacillus and pseudomonas do, but definitely most of the azos do. Plant growth promoting bacteria, here's another one with pesticides. Biopesticides, sorry. Different explanations on different areas and where the microbes are. That's inside of a microbe itself. Endophytic microbes are inside of the tissue. Rhizosphere just outside of it. And in the soil as well. They're everywhere. So those people that go and those particular who just strip the soil, go and spray and get rid of everything, all this is gone. So if you've got to go and reestablish all this, something will come in on the back of an earthworm and it'll be bad. Bang, there's nothing else around. It's gone and just multiplied so fast, bang, everything's wilting. So if you can get the biopesticides, is a good way to maintain the microbe account in your substrates. And remember too, what was there's a good story about the giant pumpkins that they have in Alaska. You know, they're the world's biggest ones, the size of, I don't know, they're massive. And they did a substrate test, and they found that a metagenomic test of the substrate, which is where you test all the microbes in the substrate. And they found them to be, there was more than 20,000 different colonies. And normally you might have sort of 6,000 to 7,000. So that's one of the reasons why it works so well, and they produced all the things that the plant needed at that particular stage, because everything was on at hand. It's like having a store open right next to you when you want to do cooking. You can just drop in, I need this, I need this, I need this. From having the different microbes there, they can just activate at the different stages to allow different things to happen. It's pretty cool. It's almost probably where I should have started. Yeah, a gram of soil has up to 10 billion cells. It can be up to 1 billion viruses in a gram of soil too, like 1 million cells of fungi. It's very complex and biodiverse. And yes, the same has beneficial, harmful, and neutral. A good soil should have an equal amount of all of them. Not be killed off from using bloody glyphosate, lemon Roundup, which is legal in Australia. It's not very good. that's a way that they can protect powdery mildew from using that microbe. Different pathogens. This is a way to prime your genes, have PR genes ready to go in defense. So if you're a breeder, you might do systemic acquired immunity or systemic innate immunity to try and activate those pathways which will code your genes and have your offspring with more of a resistance to that problem. I've done that a fair bit. It's really good and works so well. I've got a video if you're interested on root tea, and you can see from this experiment here, I did a month of testing growing teas, and from on the x-axis here is the ppm, so 900 goes up to 2,500. That's up near where it's your salinity is too high, and down here on the is hours. So when I started it and how many hours it went up in 4-hour stages up to 48, because remember when you do a tea, it's got to be enough cycling for the oxygen to go through and kill all the bad things, because you don't want to be promoting good things, culturing it for say 6 or 8 hours when all of the anaerobic stuff can still survive in it. So by doing it a longer time, you're allowing that to happen, and I think it's more than 24 hours or something like that, but that's why I go for at least 36 to 40 hours. But yeah, the root tea, so it starts off at what's that, 1,000. After 12 hours it's up at 1,300, and it's stationary for a while, then it keeps growing, which is rare. So over here, a normal phase of microbial production, or their growth rate, is it comes in, see down here, 6 hours, 12 hours, comes in, log, and exponential is when it's using all of its electron acceptors and its minerals and stuff that's available to it, and then all of a sudden it's used it all, and it's stationary, and then it goes into a death phase where it forms its endospore so it can reproduce again when it's in its favorable conditions. So yeah, you've got a lot of microbes in your roots, because from just going through this talk we've found that out, and you may as well use them. So instead of putting them into your compost heap, why not pull them out and put them in a tea first, because they're all the ones that probably would have worked really well with your plants in your environments. So by going and using those microbes that were already there and culturing them up, it's like giving it a big boost of fertilizer that's bio. So it's a really, really good bio fertilizer. Here's the colony rates that they were talking about on the back of them. Now they got here, here's one, 8 times 10 to the power of 10. So you have to do 10 plates to get the sort of colony forming units that you've established into these numbers. So yeah, if you look in the back and it's got times 1, you go, wow, there's not too many in there, because if you've only got to do a serial dilution of 1 to fill a plate, that's not a large amount. But if you've got times 10, that means there's a high amount in there, because you've got to dilute it 10 times over, 9 mils into the next one, 9 mils into 1 mil, 9 mils into 1 mil, and keep going for the serial dilution. So that's a good way to tell that the microbes in the back of your packet that you're looking at is worthy. How to read the numbers. And this is the last one, and this is a chart that I was talking about, how it shows the plant with all of the different various microbes from its different genus and species. No, it's not, it's just genus. It doesn't say the species. See, it's Bacillus species. But you have to go and see what type of Bacillus subtilis you want. But yeah, different areas in different parts of the plant, but they all exist. So if you're one of those, you think you're growing indoors and you're very synthetic, and you have to spray all the time, you're killing all these off. So when a problem happens, it's going to multiply that pathogen that's got in there from the air, or it could be jumped off your shirt that you're in there, or it could be coming from your water. Somehow it's gotten into the area that your plant is, and it's multiplied. So by having all of these organics still kept up and possible biopesticides you put in, wow, it's love and life. So there's the talk today. Hello, Ship Sunshine. How you going? Nice to see you, mate. Just been talking microbes today. Microbes, why are they good? Free microbes from the air, how you can get yourself, because remember the plant needs 95% comes from the air. Actually, I'll just show you that. Haven't been going for that long. Study soil science. I think there's one at it. Yeah, these are areas where their microbes is low, and you can see nothing cycling. So you could call it, and they would be synthetic farmers or growers that spray and kill everything. Where organic, us, look at ours. Everything is at a level which is available to the plants. It just needs those microbes to convert it. And I'll go to a better one. Please be here. All right. Looking for the levels. It says the numbers. It says 95%. Your CH and O comes out of the atmosphere. What's my other soil science ones? What was the first one of? Cell biology, disease, environmental soil chemistry. There it is. It's this one, I reckon. Uh-oh. Cation exchange, it's not even close. Far out, huh? I like showing you some numbers. It might just be in soil, actually. I'll go back to soil because it should be in here. It should be. All right, I'm just going to go through them because it's got to be on soil testing. No, we just want on soil facts. Back to the. I'm going to go right. I'll just skim through fast, see if I can see it. Because it's extremely relevant at this point. That's the end. Great. That's pretty bad. It's just one simple chart. I don't want to go into the courses. It's just to show you how important it is. By sourcing the right microbes, you can help the plant out immensely. All right, I'm going to have to look hard up because I want to get it. It's simple for me to. It should be simple for me to find. It's not even. It's in the study. What's down here? I'm going to have to pull it out after this. I'll have to go through and make sure everything's a little bit more organised, a bit better. I wonder if it's soil science. That was the first one. It's not in the rad bits. It's just got those two little bits. I think it was this course. I've got a few, plus I've also got a few charts on it as well, because there's been updated ones on it, on the amounts of these. It says the carbon, it's got this broken down a few times, because that's the amounts that you want. But I want to show you in the percent form, so you know that I'm not just reading it off the top of my head. I'll find it. It just might be. No, there's the pH, which is pretty relevant too. But we're talking about microbes. So if you can just source the right microbes, you can get the right plant nutrition. This where it is, plant nutrition. Oh, it might be under plant nutrition. I'll look under that folder next. Let's look under that. Plant growth and development. Of course it's not, it's in the cells. Oh, there it is. Oh, that's the essential elements. Yeah. Where's the number? There it is. Finally. So you can see of the 17 essential elements that a plant needs to have its growth fulfilled, there's a couple of beneficials too, but 96% comes from the atmosphere. So if you can also get your 1.5% of nitrogen from the atmosphere, from your azos, from your azotobacter or azospirillum, you've got 97.5% of the plant's nutritional requirements taken care of, and all you've got to do is water it and look for potassium and calcium, magnesium deficiencies, as an example. And if you've already inoculated that substrate, there's a high chance you won't have this unless you've got a cultivar that's a pig in calcium or potassium, for instance. And then you could just do defoliar sprays at 100 ppm to fix those. But that's an example of getting the right microbes in the right scenario where you'll be in a win situation. Let's talk in microbes. All right, there's a few, I think there's a question coming too. Sunshine, there you go. The difference between bottled nutrients and living soil is a whole food diet versus a processed food. Processed whole food diet. Yeah. Yep. You could say it's something like that. Yes, and it also takes care of the health of it too, because by not bottle feeding it, it has a chance to fend for itself, which strengthens it. So you'll find it'll have less pest and disease problem. Hey, it's Aussie Jeffro. How you going, mate? What's Aussie Jeffro say? Well, I'm trying to put a show on here, and that's, yes, I'm managing, and today's been all right. At least we've got a title, today's microbes so far. Aussie Jeffro goes to say, what is the best method or time for taking off the male parts? Oh, masculation, that's called. Take it before you see the bananas, or when you see them hanging down, or just before, or what? Off topic, sorry. No, mate, that's good. It's a question. So I'm appreciative for questions, because I can put it, I'll title today's show's microbes plus masculation. And that's removing the male bits. Yes, the anthers, they're called. And when's the best time for taking them off? You want to see it when it's in the sepal. Let's just show a bit of taxonomy. So it's not like, I think even in this section it has it. I'll just skim up a bit. I think it's right at the end. Memory. There's another one of those charts I was looking for. There it is. Here we go. So here's the bits, what they mean. So you've got the male part and the female part. So up top is male, bottom's female. So this is what it looks like. So they're called anthers or bananas, because see, they're actually dead set. They do look like a banana, and even under the microscope, they look like a banana, honest. So it's a good, word that it says there. But once that it's opened, so that it's a sepal, S-E-P-A-L, has opened, that means that there was some pollen underneath it. So that's opened already now, and it's a little bit of pollen's gone out. But once the anther opens itself, that's when it goes puff, and you'll see a big puff of it. So you want to masculate or remove these male bits in their ball form. So when it looks like a ball. So as soon as you're sort of starting to see it's got little nuts underneath it, just take them off. And honestly, there's not much to take off. So the problem with your plant is next time you want to go through, try and put it in a cycle when you know you've got a lot of females, because they'll be releasing ethylene when they're going into day 1 to day 20. They release a lot of ethylene, and that's a blocker or a female promoter. So it'll help you with your herm type of cultivars. So if I've got a herm one or ones that I'm trying to bring out its femaleness, I'll always put it in with a multi-crop in day 1 to day 20, and at the same stage when it's flipping, and then that'll help it out. And then if you do come to see them, you want to put it, you can move the plant, because if you want to sample it, you've come this far, why not? Move it to the spot where it gets less wind flow, and you can even put, you break them off. So every 2 to 3 nights, every second to third, two times a week, you'll be going in, pulling the plant and spinning the whole plant, rotating it around and looking at it to see where little balls are and just plucking them off. And it'll be right. You can let them fall on the ground because they can't mature after that. They might puff open, but they'll be immature pollen grains, and that won't be enough to fertilize these bits down here, the stigma and the style. So this is the bract, the perigonal bract. So what happens when these pollen grains, they'll land on top here, and it puts out a pollen tube, and it goes all the way around, and it wants to come down here and go into the ovule through the micropyle, and then it's, anyway, that'll do for the. So to stop that happening, you don't want the pollen grains to get on top, and you want to be immature if they do land on top. And you can tell if you can tell if, am I still talking? Am I still talking? Yes. Oh yeah, okay. You can tell if it's fertilized because the stigma, it shrivels. So after it's been fertilized, so if you were to fertilize it within 36 hours, 24 to 36 hours, so usually the next night or definitely the night after when you come in and have a look, it'll be shriveled. So then you know this bract is fertilized and it'll be producing a seed instead of trying to go and swell and multiply its bracts. Yeah, that'll do. That was answered pretty well. Thank you so much. You're welcome, mate. I've got it growing up with a girl. Oh yeah, nice. And worst case scenario, if it does, a few do open, it's not going to ruin everything. You've got to take a really big event for everything to be stuffed, because remember those bracts are at different growth stages and can be fertilized at different times. So if that only opens, bang, 1 or 2 open, okay, good, it's hit a few of them, but that's only at that particular time, so you'll only get a couple of seeds. So you're not going to go and get male plants. You think some people think there's a misconception of a few of those ruining everything. It's got to be a pretty big event and continuous event where it'll be opening every night nearly. Another few open, and then over the probably 40-day period, because it takes 20-odd days to produce the seed. Here we go, 6 minutes. It's a younger female plant. Oh, yeah. Here we go. Because it takes 20 days odd to produce the seed. So if it gets fertilized at day 45, you'll expect to see immature seed seeds when you harvest. So then you can know that it's been fertilized, you had a late herm problem. So you can go off this timeline. And the risk of viability loss, I don't know, as soon as it goes to day 30, if you pollinate, I usually pollinate about day 22, because if you do it right at the start, it slows down the vigor. But anyway, we're not into pollination. We're talking about masculation. What else do I have on masculation? I actually think I had a video for masculation. Don't know. I remember I got some slides on masculation somewhere though. But not here. That's where it should be. Seed dormancy, seed germination, no. Oh well. What's that? Seed counting. What's that? No. All right. It's good for seeds. And the microbes. Also, there's endophytes in your seeds. So that's why the seeds can't come across the borders. Because if you get your seeds from an organic source, they'll have, I'm still sharing. All right, try and find some endophytes in seeds. Under microbes, possibly. Seeds with trichomes. Just to relate the two would be pretty cool. What about into the microbe section? Anything here with seeds, biopesticides. Went through them a fair bit. No, was there any other questions? I've got my tablet open. That's awesome. It's a younger female plant. Good. So that's cool. So I'll just try and relate to both the biopesticides. What's under this one? Oh, there's heaps. Botanical pesticides? No, just under the microbes. Biopesticides. Biopesticides, yeah, slowly going up because people are getting educated. Oh, that's how people did it for 4.6 billion years before us. Oh, that goes into the how to do it. No, I'm not going to cover all this. I want to stick to the microbes section. It does touch on it though. Release the natural enemies. No, Bavaria Bastiana, I'll use that one too as a directed note. Come on, where's the microbes? I thought it did touch on it. Everything, it's chemical. Soil treatments, seed treatments. You wish to soak your seeds, it's kind of related because it's a seed and microbe topic today. So if you were to inoculate, which it's extremely beneficial, you can try all different types of things. Yeah, all it means, I usually wait till the white radical comes out, the little white tip when it germinates, it's called a radical, and then you can inoculate then. Because if you do it first, I've found, because microbes will reproduce every matter of minutes, where plant cells reproduce in hours, like 15 hours. So if you get too good of a colony that's in its too good of conditions, it'll take over. So I always wait until I've got the actual cells I can see, like the radical, you know, the plant root tip, and then I'll do it then for that reason. Because if it's taking its time, it'll, within 5 days, bang, everything else is taken over. All the bios. That's a pretty cool little tip. Biocontrol, yes. And they are rad. No one talks about them. I don't know blimmin' why. They are flamin' rad. They use 4 watts of power. If you get the LED one, don't get the tube one, the 9-watt tube, because they use 18. Get the LED, use 4 watts. And it's got some little, it even gets fungus gnats. It gets the smallest of small things as well as the big stuff. So, yeah, look for a few of those bug zappers. Online, I'd suggest, and they want them to look like this, because there's quite a few around that don't look like this, that are rot. These are rad. They're about $20 each. They're so, and that's supposed to be, they say it's a UVA tube light in it, the spectrum that's let off it. So you could use that in bloom time, which is going to give a little bit of UV input, which is a little bit of a stress, which will activate different pathways and, yeah, stimulate your secondary metabolite profile. So by sticking one of them on your flower cycle in the room, wow, more, you can nearly call it biopesticides, because you're not spraying or doing anything else, but it's just another thing that bloomin' works. Some of them come with this bug catcher. It's just a strip, and some don't, so don't be care. Oh, there you go. Ones that don't, just put out a piece of paper. It's got paper on it. Yeah, really good. Insect eggs, no, it's microbes today. Yes, if you have microbes, see that, that's a marigold in front of it. That's called a trap plant. And if you stay organic like this is, you can get results just like that. Pretty good, eh? And it's a good idea, once that, if you know you're, like for instance, you're growing indica. Yeah, look, see the size of it. If you're growing indica, you can put out this netting, which is a good idea, because you push the colas through at the start and they're going to hold its weight. See, like this big heavy one at this here? Once this gets into day 40, this is going to be swollen down, touching the ground, and it's not going to be good. But kept like that, it's a high chance. This just needs a little bit more pruning inside because you can't see through it. So I'd be worried about a bit of late season mold. Or if it rains, bang, this wants to come down before it rains, because there's a high chance it'll be above 90% humidity inside there, which is going to germinate bacteria. Where's that chart actually? While I've got you here. It's in videos to make. Bacteria. Breeding tips. Might be in here. Put the chart in here. No. Oh. It'd be fungus resistant genetics, is in that one. Trimming roots, no stress, spinalites. Maybe that one, I'll have a look. Now, of course. Bricks, breeding tips. This one. No, that's the cation exchange. There it is, Canamulch, I think. It's just inoculation results. Fungus resistant genetics. This one keep coming up. Let's have hasn't got the chart I was wanting either. They're the common fungal diseases, which is in microbes, because microbes are considered fungus and bacteria. So yeah, there's a test from doing fungus-resistant genetics, doing that in store. So it's just inquired IAR, inquired assisted resistance, inquired. It's not assisted, but it's called IAR. We inoculate a little bit, just like we get with our human. We go to the doctors, they'll give us a bit, and it's supposed to give us resistance. So I've given this a little bit, and I've tested it, and it's shown me that it does have resistance in it. This was 25 days in 95% humidity. Where's the chart that shows you the actual the humidity of the microbes? What's this one? The nutrition of the microbes. This is pretty cool too. It just shows what they need, the essential elements of a microbe. So by maintaining a organic substrate, you're going to get results if you feed your microbes that way. I'm looking for the chart for the, I know where it is. It was in food microbiology. Of course, it's none of these that I can see. Microorganisms in food. Most of these, yeah, the things in food, we're not talking about these microbes. The chart, the percent chart. I'm up to, back to here. I talked about it a bit too. I always like to talk about it, actually. So I'm surprised it's just not coming up. I wonder if it's in that section I checked before, because it's pretty important. It just shows you the humidity levels that what can germinate in. So if you've got over 65% humidity, Saccharomyces cerevisiae, it's a fungus, it's a yeast, that can actually germinate. What's it look like again? The percent chart. Might be in, it should be in microbes. What about plant science? Disease, plant problems. One of these ones at the start? Not one of these ones here. Let's have a quick squeeze. Oh no, it's just got the pH. Yeah, and also see with your low pH, bacteria reduces. Soil pH beneath 5. So not only are you losing everything, there's your essential elements I was looking for before. Another one of those charts that shows you 17 essentials. 95. Yes. There's the one before with the 1.5% as well. So if you can get that from your azotobacter, you're at a win. Deficiencies. I was looking for the percent chart. Because we're in deficiencies. No. I would not get anywhere here. That's a bit of a shame. I'm going to have to find that so I can talk about it later on, next time, because it's really important. And I remember I have talked about it in the past. Last try. Plant problems, it's not there. Rhizosphere. It's in the rhizosphere. No. Wow. Not very organised today. Water flushes, pressure, water. It's to do with water. Flushing, root precipitation. It's to do with water. Please be here. It's not here. I can show you this one, bud. With the right amount of water, you get the right results. And at any age, it is possible. Good on you, Grandma. Good on you. Freezing plant. No. Water and life. No. Aquaporins. Flushing. Okay. Plant immune. No, it's more about water. Yeah, distances to acquire resistance, because you know the plants speak to each other through allelopathy. So I did a test to see how close it was. You can see that downwind. It worked. It spoke to each other and induced a leaf curl disease, bacteria. Organic farming. No. Is that where it is? Organic farming? No, that's right. That's those examples of those tests that was done. It's a bit of a letdown. Would have finished it off quite nicely today. Just cation one. A few things into it. It's also about exchanging elements. That's a lightning one. Yeah. That's also you can get free nitrogen out of the air. It's not really from microbes, but it's from your lightning. And that's the process that it happens. Process and how it happens. Where nitrogen is zapped and mixed and blended, and the outcome is your free nitrogen in thunderstorms. It's also how they make ammonia in the lab. And synthetically, they use heat. There's that one. Where's the percents of? It's got to be on the microbes or studies. Should I keep looking for it or what? It's sort of taking a bit too long here. I've already been through that one. It's not there. Okay, last chance in studies, and it's not. Because I'm trying to find the. Whilst you think of questions too, if you've got any micro questions, particular, any question I don't mind, but any micro questions, drop them down. Nanotechnology. It was to do with the food one. Oh, that's right. Please be it. There it is. It's right down the bottom. So above 5%. What a better one? Scenario. No. But yeah, so yeasts above 60 to 65% have the chance in germinating spores, as well as aspergillus, which is very common with our stuff. And then you go up a bit further, which it's more yeast and molds. And you know, it's there to tell the difference with types of the fungus that they are. is that the yeasts are the colourful ones, and the molds have the tall-looking hyphae. So yeast are usually flat, and the molds aren't flat. So you can tell. And then the colours, you can tell the differences as well. And offhand, I can't remember. Yeah, penicillin. But yeah, above 90%, and look, all of the bacteria start germinating. So that's what I learned from this course. So that's vital. And remember, just because it might be 60 or 65% on the outside, you're reading on your gauge, inside of your chunky numbers could be 90 to 95%, which your grey mold is just thriving. Or your botrytis is grey mold, yes. So that was pretty cool. So yeah, we went through some good microbe stuff. So I can title it Masculation, thanks Aussie Jeffro, and the microbes, and how and why and what moistures that you want to try and stay away from. Yeah, oof, tough times. And also with the fans make a big difference, because fungus loves no air. So by having airflow, it really helps a lot too. So that's why with the bigger plants, you'll chop out leaves in the middle to create gaps so you can see all the way through, so the air can flow. So that's a really good tip as well. If things are getting a bit huge and out of control, you'll definitely prune it, because once you get the grey mold, you can't do nothing with it. You can't do it's really bad. I've grown up with, scoring in Australia, so I know what it tastes like. And I've smoked lots in the past growing up, because, well, you can't help it in Australia. You take what you get. That's what it used to be like. So yeah, I've known it, and it's not healthy for you. There was that chart before, actually, which showing it coming into your airways. Where was that one? Yeah, here we go. I'll show this one, because just by being around it, once it's grown, and then you'll see the puffs come out. And once the puffs come out, that's the spores. Once those spores go in, they can regerminate and start to grow and give you internal problems, like Aspergillus does. Aspergillus fumigatus. So, and Aspergillus is rife in, especially in North America in the industry. That's the highest test positives for in plants. It's so you have to be careful. So who knows? Some of us Australians might be used to it. We've had so much of it that we've got a bit of an immunity for it. Not anymore, thank goodness. Gee whiz. Try not to. Or if it does, that was the point. That if you do get it, there's nothing you can do. They've got, in licensed producers, they have to have the irradiation machines, which can zap spores and reduce the numbers. But once the grey botrytis gets in, it's all over, red rover. It's holy moly. You can't, well, once you touch it, puff, all those spores come out. So it's dangerous just for that. And yeah, so good luck. And once you, say if you were to extract it, they'll float. So you'll be extracting all the other things at the same time. So maybe if you want to, even when it goes through, they do chemical extractions, it still shows up pretty decently, because the chemicals pull out the toxins that are in those fungi spores as well, and it's horrible. So it's something that's got to be taken care of. So if you can have mold or fungus resistant genetics, keep your temperature in bloom, keep your humidity in bloom below 60%, and fans going. And if you get big chunky numbers, make sure you got air gaps and ways to deal with that. And have your sticky traps around and your card and that machine to get to zap stuff, and it'll help a lot. Just some ways to mitigate microbe problems. Because yeah, but microbes, remember, they just germinate from, they need water to germinate, which is humidity. So if you give them that, it's pretty much, if they're there, which a lot of them are in the air or on us, they got a chance in that happening. Or in the plant, there's even spores, dangerous spores in the plants that we that are around. Nothing's sterile. It's all got it sitting there. Little endospores sit there. Probably even in our beneficials that we grow, there would, that we buy, there would be even some in that. It's yeah, and just once it gets out of control, don't want to get there. No thanks. Well, there's no real questions coming through, so I reckon that was a good show today on microbes and masculation. Yeah. So bring your topics again next week, or at least bring some topics. Bring your questions to next week, and I'll try and elaborate more on the topic. So I appreciate everybody who listened, everybody who was in chat and helping out, and yeah, hope you all have a wonderful week. Thanks again, everybody. I appreciate you listening and all your ears, and Monty for coming in too late. Or he was probably already in, but it was nice to see you, Monty. Yeah, bring your questions next week. We can elaborate more. It was fun again. I hope it helps somebody. And happy growing, happy breeding, and good health to you all. Have a good week. Bye-bye. Thank you. Bye.